Assalamu alaikum. Good morning. My respected greeting to chairman and members of committee, my teachers, family, friends, and all audience. Thanks for being here. Appreciate it for your encouragement. My project title is Molecular Detection of Helicobacter pylori type A gene polymorphism and some related interleukins level in samples from patients with gastrointestinal disorders, supervised by Professor Dr. Amna Nahmatouini and consultant Dr. Basim Askar. بسم الله الرحمن الرحيم. وقل اعملوا فسيرى الله عملكم ورسوله والمؤمنون وستردون إلى عالم الغيب والشهادة فينبئكم بما كنتم تعملون. صدق الله العظيم. In this presentation we will go through these outlines and we will start with introduction and aim of our study. So let's take overview about the anatomy of stomach and the main part of this organ, which is located between esophagus and small intestine. Start with the duodenum. The proximal part is called cardia, while the upper part is fundus, and the part between razor curvature and laser curvature called body or corpus, while the lower part is pyloric antrum. And let's remember this place because our main sample have been isolated from this side. So in the beginning of 1918, the gastroenterologist Barry Marshall and the pathologist Robin Warren have been found association between gastritis and the human gastric pathogen Helicobacter pylori. In 2005, for this discovery, they received the Nobel Prize. These are gentlemen on the left side, Dr. Marshall, and the right side, Dr. Warren. All the respect for Dr. Marshall because he put himself at risk when he drank the broth that contained H. pylori to prove his findings. So Marshall and Warren discovery founded the concept that H. pylori colonizing the gastric mucosa and can lead to a variety of other gastrointestinal disorders like gastritis, peptic ulcer disease, and also gastric adenocarcinoma. Since 1994, WHO classified H. pylori as class 1 carcinogen because of its relationship to gastric cancer. So how this bacteria can survive in the harsh acidic environment like stomach. As we know, the pH of stomach is 2 and it's reached about to 6, which is alkaline environment, by the action of urease enzyme that produced from bacteria, which convert the urea to carbonate and ammonium. And this is the main reason of alkalinity. The GAG-8 toxin, also the main toxin, translocated by using type 4 secretion system inside the epithelial cell, and that will affect by direct or indirect way on transcription of pro-inflammatory genes like cytokines and interleukin-1 among them. The VAG-A, which is the main toxin in our study, which related to vacuoles formation and literally will cause mucosal injury. The aims of our study: number 1, investigation of VAG-A genotype from endoscopic specimens of some Iraqi patients and study their association to clinical outcomes. Number 2, detection of H. pylori GAG-A, ISA, and their prevalence with specific H. pylori VAG-A combinations. The synergistic action between ISA, GAG-A, and their effects on different pathological groups, and the similarities and differences between local isolates and global isolators. Also, we have a silico study for VAG-A S1 region and the importance of its amino acid sequence within this region, determination of interleukin-1 beta, interleukin-8 level, and their correlation with gastrointestinal disease. And then last, we have estimation of interleukin-1 beta polymorphism within 5' untranslated region and their relation to gastrointestinal disorders. Now let's move to subjects, materials, and methods. So by this flowchart, we will explain our main steps for this work. In subjects, we start with the 2 groups. We have 210 patients enrolled in this study, and also we have a healthy group. So blood sample collected from patient, serum was isolated for anti-H. pylori immunoglobulin G detection. And for healthy and patient, the blood sample have been used for detection of interleukin 1 beta polymorphism at promoter region, we have two sides, minus 511 and minus 31. By Sanger sequencing, then the result have confirmed by reflex PCR. In serum was used for measurement of interleukin 1 beta and interleukin 8 level by ELISA technique. An antral biopsy collected from patient group only. 1 biopsy for rapid urine test, 3 to 5 biopsy for histopathological examination, and 1 biopsy for molecular detection of H. pylori. After obtaining the result of molecular detection, we have 120 as a positive result, and the negative result was excluded. So for this positive result, we start for detection of virulence factor genes. Number 1 with BAG-A genotyping, and second gene GAG-A, and also ICE-A alleles. So what the principle of rapid urease test and how this test will work for detection of H. pylori. The sample or biopsy are collected by using upper endoscopy, and directly the sample are put inside the test tube which contain urea agar. If that biopsy colonized with bacteria, so the urease will produce, and this will hydrolyze the urea to ammonia and carbon dioxide. Then the ammonia will react with H2O to produce ammonium and hydroxyl group, which is the main reason for alkalinity. In molecular detection, we start with DNA extraction from the biopsy by using Kiam spin column from Kia gene, and the measurement of DNA concentration by nanodrop from Thermo Fisher and by qubit from in vitro gene. The qubit is instrument, it's a 4 meter instrument. So it have two standards, one with the lowest concentration of DNA and second with the highest concentration. These standards help for creating the standard curve, and by this standard curve, our unknown sample will be detected. In molecular detection of H. pylori for 16S ribosomal RNA, we used 4 different sets of primer. Set number 4 was newly designed in this study by Genius Prime in cooperation with the Primer 3 Plus parameters. And for reason, we have choose set number 4 to work together with GLM gene in a multiplex PCR detection for this bacteria. In detection of virulence factor genes, as we said that we have 3 main genes in this bacteria. So this figure explains the main polymorphic region of BAG-A gene, which have S for signal region, it will be S1 or S2, and I for intermediate, it will be I1 or I2, while M for middle, it related to M1 or M2. This figure explains the primer binding site that we used in our study. 2 primers for S region detection and 2 primers for M, while for I region we have used 3 primers, 1 forward and 2 reverse primers separately used. And the quantitative measurement of interleukin 1 beta, interleukin 8 and interleukin 1 beta by using Abcam ELISA kit. And for detection of polymorphism at promoter region, we have used 2 primers to combine or to involve our 2 SNPs, which are located at promoter region, between of them. So this is forward primer and this is reverse primer. This is minus 511 polymorphism and minus 35 high fidelity DNA polymerase. This as a new generation of DNA polymerase with ultra low level of error, and this polymerase used for sequencing issues. And loading about 5 microliter from PCR product into gel, then choosing the samples with sharp bands, and sending about 45 microliter of from amplicon to macrogene for standard Sanger sequencing, post-trans, forward strand and reverse strand. Then the analysis of results have been done by Genius Prime software. So after receiving a result of these SNPs, we found the minus 31 polymorphism was effective in our study. So we confirmed the result of Sanger sequencing by using another technique, which is reflex PCR. Before going to lab and use the enzyme, we checked by in silico study if there is another recognition site for this enzyme. This is forward primer and this is reverse primer, which are different from primer have been used in sequencing. and we found there is only one unique site for allowen enzyme. Then the result of virtual gel electrophoresis are showing by this gel, virtual gel. The list of primer used in this study. Now in section number 3, which is related to our results. Distribution of patient according to endoscopic findings. Once we collected the report, endoscopic report from the gastroenterologist, we got different lesions, but the highest record or highest frequency with gastritis, it's 95 out of 210. Then the result of rapid urase test, duodenal ulcer was highest sensitivity with duodenal ulcer samples, which have been detected in 20 out of 21 samples, followed by active gastritis, but the lowest sensitivity with gastric cancer and intestinal metaplasia. Then for molecular detection, we got a concentration for DNA ranged from 5 to 120 nanogram per microliter. As we said before, 4 sets have been used for detection of 16S. So the result of set number 1, it gave us positive results for all samples. So at this point we got a doubt regarding to this primer. Once we checked the primer by using primer blast, we found there is a high similarity with another microorganism. For example, this is Campylobacter fetus, and we can see it's bind with the genome of Campylobacter fetus. That's not given us a result or good result. That's why it's excluded from study. And for set number 2, set number 3, the problem was different. These primers had given a non-specific band at negative result. This non-specific band could interfere with our analysis. That's why those are excluded also. With the primer, which is a newly designed, there is a good and sharp band, and for this reason, we choose this primer to work together with GLM gene for multiplex detection. The high sensitivity with 16S ribosomal RNA detection, which is 97.5, while with GLM gene it's 69.1, because this was detected in 85 out of 120 as a positive result. The result of standard sequencing, this is our local isolate sequencing, and once we did the multiple alignment with the global isolate, the phylogenetic tree was constructed. This is our local isolate, which is totally similar to isolate from India, and it is also similar, more similar to Vietnam, South Korean, and Japanese isolate, which is Asian isolate. And it's going to be far from Russia, USA, and Germany, and also African isolate, as Angola had been represented here. This tree, sorry, it was constructed by neighbor joining tree. 10 sample was deposited in the gene bank. The accession number has located between 22 and 20 and 11. So we chose IQ for abbreviation of Iraq, and the date of submission is 1st of October last year. The name, our name is there, and the isolation source is from gastric mucosa. We said that we start with 210 patients, but after molecular detection, we have 120 as a positive result. So the demographic study was being analyzed for only the positive result. We found there is a significant association between age and infection, and the most affected age is between 40 and 59 years, while in gender there is no any association. In residency, we have 72 cases in urban places, while 48 in rural. And the result of some virulence factor genes. In VAG-A combination, we got all possible combination except S2, I2, M2, which is not present in our study, while the highest rate with S2, I2, M2 is 50 out of 120. The multiplex result for S and M region, the lowest band related to S1, while second band related to S2, and third band for M1, and the higher for M2. For I region, it's not possible to go through multiplex PCR because the product size close to each other. The I1 is 426 and I2 is 431. So we did a separated reaction for that. The result of sample number 1 and positive control sample was positive to I1, and these samples are negative to I2. The rest of sample in this figure, positive to I2 and negative to I1. The good result we obtained from detection of S region, all 17 cases of gastric cancer was being detected with S1 region. And for VAG-A mosaic or combination, these 17 cases are carrying S1, I1, M1. So due to this importance of S region, we prefer to analyze this region for in silico study, and by focusing on the amino acid sequence. There was a previous study by McLean and his colleagues had been detected 3 tandem repeats of amino acid within S1 region, which is glycine followed by 3 variable amino acid, then glycine again. This was repeated 3 times. And this repeats located between residue number 14 and residue number 26. While these repeats are not present in S2 region, as McLean have been saying. So, for visualize this region, we download the FASTA file related to citrane carrying S1 and another FASTA file for citranes carrying S2. We did the translation, our virtual translation, according to bacterial genetic code for this nucleotide, and for annotation of the position, because VAG-A normally going through 2 proteolytic cleavage. So we took in our consideration this point, and we found yes, there are hydrophobic tandem repeat between residue number 14 and residue number 26, while at the same region of S2 citrane it's not present. But after focusing on the sequence of S2, we found the same tandem repeats are also present in S2, but it's going to be a little bit shift forward due to insertion of 9 amino acid at N terminus. As we can say, this is the sequence of amino acid for S2. There's nothing like a tandem repeat between 14 and 26, but it's located now in S2 between 23 and 35 due to insertion of amino acid. So we search for another reason why this toxicity and high effectivity for S2 toxin. We predict a secondary structure for both toxins, and for S2 we found there is a coil structure at N terminus followed by turn structure, while for S1 start with 2 amino acid with alpha helix and followed by beta plated sheet. So beta plated sheet and alpha helix can give us a strength and hardness for this toxin. We haven't stopped at this point. We moved for production of tertiary structure by using expositor and the Swiss model through blast for searching about evolutionary related structure. There are 5 tablets of tertiary structures are predicted, and the one with the highest coverage and identity have been chosen. This structure detected by Zank in the last year through cryo-electron microscope. This is the 3D tertiary structure. This is C terminus, and this is N terminus. S1, it's located in this region. So we did alignment with our target. There are 26 amino acids are missed or failed to detected under electron microscope. The reason is due to the high flexibility of this region. But before that, we predict the secondary structure of these missed amino acids in our study. The result related to GAG-A, it was detected 71 out of 120 positive results, and there is no significant association with our pathologies. For ISA1 and ISA2 detection, in 18 cases out of 21, for duodenal ulcer pathology, there ICE-A was detected, while ICE-A2 detected in 8 out of 9 cases of gastric cancer. So from this table we can see ICE-A1 related to duodenal ulcer, while ICE-A2 related to gastric ulcer, and both of them are associated to peptic ulcer disease. The result of cytokine study through ELISA, the level of cytokines for both interleukin 1 beta and interleukin 8 was higher in patients than healthy groups. The highest concentration with gastric cancer for interleukin 1 beta, while no big differences between gastritis and intestinal metaplasia. For interleukin 8, there was no association with our pathologies. The result of -31, which is cytosine replaced or substituted by thiamine, and the result of sample number 1 in this figure, there is one peak at the site of polymorphism, which is related to cytosine, and the genotype is CC, while for second sample, 2 peaks at the site of polymorphism, one for cytosine and another for thiamine. The genotype will be CT, and third sample, only 1 peak related to thiamine, the genotype will be TT. Let's notice here, one cytosine have been replaced by thiamine. The Tata box will initiate at this region. Tata box as a recognition site for related to gene expression, and this is the start codon of interleukin 1 beta. The result related to -511, which is thiamine replaced by cytosine, and sample number 1, only 1 peak related to thiamine, the genotype will be TT, and for sample number 2, there are 2 peaks at site of polymorphism, one with the green color and second with the blue, one for cytosine and another for thiamine. The genotype is TC, while third sample, only 1 peak related to cytosine, and the genotype will be TC. We found that patients with severe inflammation, they carrying T allele at -31 region. And for this importance, we have confirmed the result of Sanger sequencing by using another technique, which is reflex PCR. The band after treatment with LO1 enzyme, the single band is related to CC, and 2 band related to TT, while 3 band related to CT genotype. Now in conclusion, number 1, the result from recent study shows that the Helicobacter pylori cause different clinical complication, and the severity of infection range from mild to high severe cases, and sometimes reach to gastric cancer. The molecular detection method of H. pylori by using 16S ribosomal RNA gene shows high sensitivity and specificity for this pathogen, followed by histology examination. The using of GLM gene in detection of H. pylori was not suitable, and false negative results in some samples are recorded. All H. pylori isolates are positive to VAG-A gene, and the samples with VAG-A S1 M1 combination are always carrying I region, while the strains with S2 M2 are detected with I2 region. It was found all positive samples diagnosed as a gastric cancer are carrying VAG-A S1 I1 M1 pattern, whereas other cases in this study composed from multiple variants. By using different bioinformatics softwares and with the help of XPASI tool, the reason behind the high toxicity of VAG-A S1 toxin in compared to VAG-A S2 was predicted. It was found that the secondary structure of N-terminus amino acid in VAG-A S1 toxin start with beta strand, while there is a cold structure at the same region of S2 toxin. Last point in conclusion, the H. pylori patients whom carrying -31 T allele within interleukin 1 beta gene are more susceptible to develop severe inflammation. Lastly, in recommendation, the scientific cooperation between researchers and MOH to obtain accumulative data recorded from archives and design the meta-analysis study for this infection. Using another housekeeping gene along with 16S ribosomal RNA for detection of H. pylori by multiplex PCR technique. Number 3, specific studies should be conducted on patients with severe inflammation such as intestinal metaplasia, atrophic gastritis, and gastric cancer as the main group. Also, high number of samples are preferred. The positive result of BAG8 gene in all H. pylori isolates lead to use this gene in manifestation of infection and in the severity of infection at the same time. The role of interleukins is important in the circumstances of infection, and the concentration of these cytokines are better to determine by reverse transcription, quantitative PCR, or immunohistochemistry technique after taking the sample from the lesion itself or surrounding tissue instead of collecting sample from circulatory system. Last point, similar studies may be conducted with another H. pylori virulence gene like DOPA and OIP gene. Thank you.
شكرا جزيلا.